Hi, my name is Andrew Cairns, and I'm a medicinal chemistry trainer here with WCAIR at the GDU. Welcome to another edition of our In Conversation With series. Today we're looking at the role of chemistry, the drug discovery process. So chemists are involved throughout the pipeline, but of course chemistry is a broad field with lots of different specialisms. And if we think of any one role, we tend to think of discovery chemists designing and making small amounts of things to test. So today we're going to learn a bit more about some other roles with the help of a few guests. Both are happy to take questions, so please submit these via the Q&A panel. Okay, so first I'd like to introduce Chimed Jansen, who is a principal computational chemist at Chimed, and he previously worked here in Dundee. And he's one of these chemists that you can't find in a lab. He uses computer modeling to identify starting points, analyze results, assess strategies for a drug discovery program. And then giving us another side of the story will be Fritiof Ponten, who is currently CEO of Biotech Fluidics, but formerly head of early scale-up with AstraZeneca in Mölndal in Sweden. Fritiof has lots of experiences in the challenges which occur between a molecule being discovered on a tiny scale and being made available for trials and manufacture. So really now I just want to each of you guys, what led you down your respective career paths? So I guess we'll start with Jimmy since I introduced you first.
Sure. Yeah, so I think when I started my chemistry path, I had no idea that there was such a thing as computational chemistry. It's really one of those fields that you just become aware of during your studies, and it combined two things that I liked a lot, so chemistry and computing. And I think probably from the first project I did, I thought that it was one of my favorite areas, and I guess through the guidance of the tutors during those courses, it really piqued my interest and kept me interested, and I followed it, and it became a larger and larger part of what was originally a chemistry degree, and eventually, you know, in my PhD, the largest portion was dedicated to computational chemistry.
Okay, and how is your sort of professional career then progressed from that point?
Yeah, so following my PhD at the Vrije Universiteit in Amsterdam, I came to Dundee, and I spent 5 years there as a computational chemist. So it was really a continuation of many of the same activities as I was engaged in my PhD, and probably a slightly more, yeah, I guess a project-oriented, team-oriented approach. And then after my time at Dundee, I moved to Chimed, and yeah, I still work in a very similar fashion, although it's not an academic institution. Yeah, the format of drug discovery is quite uniform, I think, across different yeah, organizations, whether they're academic or commercial. You basically have the same teams, and in many of those teams, you'll find a computational chemist or a partial part of the time of a computational chemist.
Okay, thank you. And I guess I'll go now to Fritiof. Do you want to describe your path through chemistry to us?
Oh, thank you. Yes, I did my PhD at Lund University in southern Sweden. And after ending that, I had the opportunity to join AstraZeneca in Gothenburg or Mölndal as a medicinal chemist. So I started off working with drug development in antiarrhythmic field. Managed to be actually introducing combinatorial chemistry in late stage lead optimization, and we were able to choose the first candidate drug that came out of a combinatorial plate in that project. As often in this business, you counter your success in how far it reached, and it was not stopped until the transition between phase 2 and phase 3 due to side effects. So it was in many ways successful. I continued for some more years until I was told that Fritiof, you should consider to take on the role to build up a scale-up facility here in Mölndal. And I'm not an engineer, but I'm practical. So I was first kind of surprised and started off a journey that I then run for 10 years, building up a facility to produce the first larger batches of candidate drugs. And in the AstraZeneca nomenclature, it was called Campaign 1. You can call it different in different companies, but it's the same thing. It's the last batches of non-GMP material. And it's used for toxicology studies, typically in rat and dog, but it could be other things as well. In amount, producing up to 0.5 kilo of API. Active pharmaceutical ingredient, I should say, not just an acronym.
And I guess while we're clarifying, I should just say at this point that might sound like a bag of flour to non-chemistry attendees, but it's really a great deal of material for a synthetic chemist.
Yes.
Okay, so I have, if it's working, yep. I have a slide with a very simplified schematic of the drug discovery pipeline here. And you can certainly find dozens of different versions of the same thing. But I've chosen a pretty simple one. And there's the bit that is kind of R&D I've put in a red box. And so I want to ask you both how you see your role and where do you fit in this process? Where do you come in and where do you bow out? And I'm going to go to Chimed first again because you come in earlier in the process, I believe.
Yeah, that's right. I would say hit generation is already an area where computational chemistry can have quite a bit of impact. Probably not in target ID or validation. That's really, biochemistry, biology. But yeah, once you get to hit generation, that's, you could do a virtual screen, for instance, to try and come up with hits. And then it continues in lead generation and optimization, particularly optimization. There are a lot of methods you can apply there to try to increase the potency, usually of a series. And I guess also for lead generation, you have to use some techniques like scaffold hopping to try to come up with new leads for a project. But that is more or less where the input ends. I mean, you do have modeling of kind of more preclinical properties, but it's a different kind of modeling than I do. So for my type of computational chemistry, which still is very much focused on how the small molecule binds with a protein in most cases, it's really hit generation to lead generation and optimization.
Okay, and you've already mentioned that it's quite a team activity. So, what does that look like, and who do you work with?
So the team will generally be, like a team leader. So that's usually a medicinal chemist with a significant amount of experience to lead the project. And then a number of medicinal chemists to synthesize compounds, and a computational chemist or part of the time of a computational chemist. And usually if the project has any possibility to have structural support, there might be a crystallographer or access to crystallography as part of the project. And then alongside that, you need all the assays that will be run. So there's a lot of biochemistry, but when it comes to designing and selecting compounds for synthesis, it's usually between the medicinal chemists and the computational chemists, and perhaps some input from crystallography.
Okay, and would you expect to stay with the project you're working on through this kind of from early hit generation through the kind of lead optimization? Would you stay with the project that time?
Ideally, yeah. I mean, I think those are the most interesting projects to work on, because then you see the whole process kind of from early stages right through. So I think that makes the project a lot funner to work on, because you kind of know that what you're doing now will have an impact further down the line, and you're much more engaged with the project. If the project is kind of passing on to other groups or something after a certain stage, you don't get as much engagement with the project and the team, simply because it's shorter, and your end goal is much closer to hand.
It's nice to stay involved if you can.
Yeah.
So, I guess you sort of bow out of, in those projects you get to stay with it, you bow out of the project about the same time as Fritiof is coming in. So, Fritiof, do you want to tell us about where you see yourself in this pipeline?
Yes, the classical view of it is that it's a process, developing processes. However, that is not the entire truth if we are looking in a drug development project like this, because any project needs to be able to supply material for their tests. Early on, 10 milligram, 100 milligram could be sufficient for everything, but the further a compound progresses, the more material is needed. And you need to, if you know already from the beginning which projects are the winners, it would be very easy, because you would just throw in massive resources there. But we don't. We know that a majority of them will drop out, especially from, let's say, a late lead optimization until phase 1. We have a massive dropout. So you always need to be slim on resources and work on the most challenging things. So in Mölndal, we practice a procedure that we call proactive work, looking at the project coming and to see where do they have their real chemical challenges, so that we could start to work with them up front, maybe as advisors for the medicinal chemists, but sometimes to develop a step here and there so that it worked. And that gave us the opportunity to have a taste on the chemistry. Because when we get to the point where the project finally decides to lock on 1 or 2 compounds, you start a clock, and the clock is running towards first time in man. And you have it all the way up from upper management in the company. No one wants this time to take more than a few minutes. But that's not the truth. So we need to squeeze things together and optimize the use. So I start to do work in late lead optimization to enable the transition. What we say here is preclinical, enable animal studies, but also studies on solid phase properties, crystallization, in what form do we want the product, so that we in the end can reach a first time in man within reasonable time.
So you're trying to balance not wanting to commit resources to projects that have a high chance of not going anywhere with being prepared to step in as soon as the opportunity comes up.
Which means that I am working with a lot of different disciplines here. Obviously with a project that has made the chemistry before, but also with the toxicologists to schedule the different screens or the different studies. And you could have a small study like an Ames test needing less than a gram or 100 milligram, but it can be a showstopper very quickly to a compound. So if we enable that early on, we could pull out the compounds that won't do it. And then we have the formulation people. You need some kind of a formulation to test your compounds. So we work very closely with them as well. And then to take the next step towards phase 1, where you need GMP, good manufacturing procedure material that is qualified for work for testing in human. Well, that's another team, and we need to keep them informed on what we do and what we don't do. What remains to be solved for them.
So I actually have a really great audience question that's come in, so I'd like you both to weigh in briefly. And that is, well, I guess it's a 2-part question. What is the standard timeline for the drug discovery process? And is there anyone that stays with the project from the start all the way through to, say, FDA approval?
Yeah, so I'll cut in because I guess I was talking first, but I don't know if there's really a standard timeline because projects are so diverse and they face really different challenges. So I think people always have an idea of the timeline that they would like to achieve for a project, but I don't think any project really progresses without facing unforeseen hurdles. So I would guess that, you know, we talk about maybe sort of 2 to 6 years for the first 3 steps, maybe with some preclinical studies. It's a wide range, but some projects are easier than others. So if you're starting with a lot of information and you're starting with an assay that's, you know, already, well, either you have a validated assay from previous studies that have been done on the target, or you just, the first one you try happens to work really well, then your project can progress relatively quickly if there aren't other hurdles. I think, yeah, that first box can really take a lot of time if it turns out to be challenging. And as far as the steps that I'm engaged in, yeah, probably if things go smoothly, a couple of years, you know, even less than that is possible, but usually it would take more time because things crop up that that you haven't foreseen. What tends to be an issue is that, as you change the properties to optimize interaction, the new properties of the compound, so let's say that you've made them basic because you wanted to have a charged compound which would be perhaps more soluble, then you find out that turns out to give problems for development, perhaps toxicity or some other challenge, and so you're constantly trying to balance different properties of your compounds to meet all the needs for a compound to progress further.
Okay.
And maybe just to touch on the second point, which the time.
The whether MD stays with the project.
Yeah, whether anyone stays, probably not, because I mean, I've only ever worked in groups which are focused on the part in the red box, the drug research and development. So that is definitely from the projects I've worked on, cut off from the clinical trials side. Perhaps in really big pharma, you would see the full trajectory with somebody who has some connection to the project being engaged throughout, but in teams that are really focused on the first half of the process, you would only be there for the first 4 boxes, and really probably the first 3 boxes, depending on how the project is being
the skills required are quite different.
Yeah, for sure.
Fritiof, do you want to weigh in on this?
I can agree. I know people that have followed very long up into clinical phase, but that was in a growing organization, and it included career swaps on the way. So we should remember that besides the process and manufacture, there is not much chemistry in clinical phase. Though there is work with solid state properties, formulation, and so on, and you could have a medicinal chemist switching career, but not regularly from a competence perspective.
Okay.
When it comes to times, we had a goal at AstraZeneca to progress a project through lead optimization within 18 to 24 months, and that's the pink box here, or that includes a bit more. And the blue one, preclinical and animal studies. Well, we usually delivered our first half kilo of material within 9 months from decision or from a go. And then you have toxicology studies, which typically take half a year. And depending on the risk awareness, if you are willing to take the risk, you could kick off the next batch early on and gain time. But most usually, you wait until you have your result and then you lose time. So I would say that this preclinical animal studies easily take 2 years, and it could even be a third in reality.
Okay. I hope that answers the question. I think that was quite a good detailed answer from both of our guests. I have another question in this section before I want to change gears a little bit. And I guess the question for both of you is, you're used to working in environments where you're sort of part of a multidisciplinary team. What do the other roles that you work with misunderstand about yours?
So I guess I'll start again. I think for computational chemists, when you are part of a team that that perhaps, you know, a team of medicinal chemists who previously didn't work with computational chemists. I mean, I guess that in itself is not such a likely scenario these days, but I guess if people aren't that familiar with the role, some of the misconceptions they may have would be about how predictive the modeling is. So what the computational chemist does is try to build models that values usually about the activity or potency or binding of a compound, and then possibly other properties as well, like the basicity or perhaps other polar properties or related predictions. And I think that there's quite a lot of uncertainty in the models. That's just an inherent part of modeling. You don't reconstruct reality. You just take what you can model, so the simplified version, and use those models to come up with predictions. And yeah, I think the misconceptions come in where people either will assume that the modeling is correct, is actually 100% accurate, or the modeling is just not worthwhile. And we get that reaction, I guess, from people who haven't had a lot of exposure to computational chemistry, so they may have one experience that was either negative or positive, and then kind of assume that computational chemistry can repeat that or will always repeat that.
Do you know how uncertain a calculation is going into it?
Yeah, that depends a bit. So yeah, there's an underlying experimental aspect to modeling. So basically, we take as much experimental data as we could, and then we use that to extrapolate or use other modeling techniques to come up with predictions. And so we have a strong sense of how far we're deviating from the experimental. And if we assume that the experimental is correct, then we have a decent idea of how predictive our models are. But it will vary very much from one model to another.
Great. Thank you. Fritiof, what do people not understand about you?
I think one thing that is commonly misunderstood when it comes to early scale-up and process development is the role of optimization versus understanding your chemistry. Because there is a lot of methods to optimize things, especially computational chemistry can help you with a lot of experimental design and so on. But what is most commonly forgotten or misunderstood is that the core of it is chemistry. What is happening in your reaction? Understand it. What is happening more than you were expecting? Yes, I have my reaction I want to see, but I also have the side reactions, or I have the lack, the final getting the reaction to completion, leaving a starting material that can be tricky. So when it comes to getting a robust and reliable synthesis, because you need a reliable route, you need to understand your chemistry. So I was often the naughty guy that asked the chemists for the crude NMR, the crude LC, because I want to see what happened more than the expected, because that is what tells me what's going on in the flask. And people were proud and really joyful to show an excellent, nice HPLC chromatogram with one peak. And yes, it's very nice, and they have done a fantastic job. But it's the crude that tells me how was it after the reaction. So
you're going around asking all those poor medicinal chemists for their crappy NMRs, the ones they don't want MD to see.
yes.
Very good. Okay, just in the interest of time, I'm going to move on now to asking a bit more specifically about the chemistry in your role, because I wouldn't want to deprive either of you the opportunity to actually talk about that kind of thing. So I guess the first question is, what is the kind of most important thing you're trying to achieve in a technical sense in your work?
Yeah, so I guess the most important thing that we want to achieve in computational chemistry is a enrichment. So basically, the idea is that the chemists eventually will be able to identify the most potent compounds that they would have synthesized while synthesizing fewer compounds. So, you know, if it would take 100 compounds to identify a compound with the right properties to push forward, if you can bring that down to, say, 50 compounds or 25 compounds, that's really a large achievement for computational chemists in a project. And I wouldn't say it's always successful. You know, sometimes you still have to make the compounds to be sure that, you know, the model is really predictive, or sometimes you know, you can only really use the model to better understand why certain things are happening, and then, you know, eventually use that in a predictive sense. But sometimes using computational chemistry predictively from the get-go just won't work. You need some time to kind of understand the series, and
you need some data to build your model.
Exactly, you need to build up data. Yeah.
Okay. Fritiof, what are you trying to achieve? What's the overarching goal?
Well, one key thing for me is to have a reliable delivery plan, because it will affect, if I fail in delivering, if I have a lost batch, that will affect the first time in mandate, that will ruin the whole planning. So what I need to develop and assure is that I have gathered the information, because it's not available. I will have to create it. The information, the knowledge, the confidence and understanding in what can I achieve with the chemistry I have. So that is from the perspective of delivering what I'm expected to do. At the same time, I need to be able to do that in a safe way, because we could have intermediates that are not at all nice compounds. We could have an end compound that is hazardous from an energetic point of view. And it, in its most simple form, an energetic compound decomposes when you isolate it and it's lost. In the most complex situation, it decomposes very violently, and your equipment, your hood, your lab, your building, your life is destroyed, and you can't have that. So you need to follow this safety in terms of energetic properties as well, so that you can do it safely. And this is bloody serious. And I can tell you that your staff won't like to go in and do a piece of chemistry that they feel unsafe with. There is a rule of thumb in explosives. 1 finger, 1 gram of an explosive, 1 finger. 5 grams in hand that is lost. And we are working with hundreds of grams. So that is a no-no. It should not even be close.
So you're really at the stage where mistakes become a problem with these materials,
yes.
Much more so than the earlier discovery.
Yes, because if you're working with less than a gram, you can handle it. You need to change your underwear, but you will survive.
Okay, I'm gonna get an audience question in here. I think it's one you can both probably give a very short answer to, but I think it's one that it's good to have an answer there for. What happens when a drug gets halted due to side effects? Do drugs ever get scrapped and not worked on any further?
Yeah, I think that happens all the time, especially at this early stage. Yeah, I think the statistics on how many compounds enter the process versus how many drugs come onto the market are fairly astronomical, really. You start probably, yeah, it depends, of course, how you look at things. If you include hit generation, which can include some very large numbers, the statistics look very skewed. But if you take the compounds that go into lead generation and optimization, you're talking about hundreds of compounds, and you have a very slim chance of making just one drug from that. So the odds of any particular compound making it through the process are very, very small. So they do fail all the time. And it's important. And I think a key thing that is often said in the industry is fail fast and fail early, which is what we are trying to do, is try to figure out which compounds just won't make it as soon as possible.
I can just agree, and especially with make the critical experiment that will kill your compound quickly, early. But I can also say that, and of course, those compounds that has been made, I have personally delivered 0.5 kilo one day, and the next day I have announced a result from the project that kills the compound. And of course, you have 0.5 kilo sitting on the bench. It's sad. But also intermediates and etc. are very valuable tools because they nurture other projects that you suddenly have. And that's one side where you can become quite popular as a scale-up chemistry in a medicinal chemistry environment, that suddenly you are the guy that has 25 grams of an advanced intermediate sitting somewhere around. That can make you really popular, I can tell you.
Okay, I'm going to I'll have one more sort of question on the chemistry roles then, which would be to ask each of you to give an example of a real problem you've encountered that you think is interesting.
Yeah, so
tell
I think
us a story.
yeah, I mean, I guess it's come up in a couple of projects, but one of the problems that I've seen, I've both in Amsterdam at the Vrije Universiteit and in Dundee at the DDU, I was working on neglected diseases, and so you're dealing with parasite targets, and then you want to find compounds that kill the parasite but don't harm the human. So the key problem that you're always faced with is selectivity. You have proteins that look like proteins that are also found in humans, and the proteins in the parasites are your targets, and the proteins in the humans are your off targets, and you need to develop selectivity, and yeah, in projects that I've worked on, trying to induce some selectivity within a series has been a key problem that was faced, and that the way to overcome that has been to usually occupy a part of the protein binding pocket, which is in some way different between the human and the parasite protein, and form some new interaction which allows selectivity to evolve. And yeah, I would say that that's in that particular area, that's a common problem that you face.
Okay, and I guess since the proteins are dynamic, that's not necessarily simple.
No, yeah, I mean, there's a lot of factors to the problem. So yeah, the fact that the proteins are not dynamic, are dynamic rather, means that, you know, certain pockets may appear to be only found in the parasite, say, but actually the human protein can fold a little bit to adapt to fit a compound that appeared not to fit in the static crystal structure. So yeah, that is certainly a part of the challenges with selectivity. And I think another common issue is that you tend to then make your compound bigger to occupy this new pocket, which has a negative effect on the properties of the compound of the series. So the chances of having a developable compound at the end of it start to fall away if you've added too much molecular weight.
Okay, and Fritiof, do you want to give us a story of a problem that you've encountered?
Yeah, a frequent problem or challenge is to set the synthetic route for scaling up a drug. Obviously, the project has made the compound in some way, but what you frequently want to achieve is that you want to be able to do your derivatization or your you want to explode your diversity in the last steps, so you can make some building blocks, and then the computational chemistry guys, they see an opportunity to make a lot those variations. That is not necessarily the best way to do the compound when you're just heading for this one. So at this stage, as we see on this picture we have up here, in the box, at least in the first 3 parts of it, we are working with a number. It could be hundreds, thousands of compounds, but now we are focusing on one. So at the same time as we are under time pressure, we also need to consider the best way to do it, to use our chemistry knowledge. And that is on the fly, so to say, because you won't have the time to do the experiments. That can be quite challenging to explain to the project that, well, you have done it in this way, well done, but we will need to change. One project, or rather my last project at AstraZeneca, the project were working in one end of the molecule, which meant that they made everything else and they did the variations on that end, which also meant that they started the whole sequence with a really expensive in effort, difficult to make chiral 7-membered azepam ring. That was really a nightmare to make. And you include that in the first step and you lose in all your losses all through the synthesis, you make losses of it by swapping that. So we did all the other things first and introduced it in the final steps. We could reduce the need for that component to 1/3, which not only meant cost, but time, and time is more of essence here. Because we were not able to make that much of it and especially not so that we could start the sequence, because then you needed it in the very beginning. In this way, introduce it in the last steps. We could work on that one at the same time as we did the other things.
So, sorry, we're getting towards the end, and I really want to get to the last thing here is just to ask you guys a bit about the future and where you think drug discovery is going and what's exciting you. And I'm going to actually start with an audience question here, because one of our fantastic audience members has hit on a question that I think is very relevant to one of the topics that I know that Jimmy would like to talk about. So the question is, how much of a game changer is AlphaFold's protein folding?
Yeah, I think that's definitely an interesting question. I think one of the issues in drug discovery is that we don't know what whole proteins look like. So there is something called the protein data bank, which contains crystal structures of hundreds of thousands of proteins by now, but it's not all proteins, and certainly not all human proteins. Many of the structures are of proteins that are in animals or insects or bacteria, where the protein is easier to express, and so yeah, to kind of fill that void in our awareness of how proteins look. Projects like AlphaFold, which predicts the folding of proteins, so essentially, eventually the structure of proteins, should be able to start to fill that gap. But the challenge there is really being able to predict the structure of proteins which are entirely novel. So if you use artificial intelligence, which AlphaFold does, you can access a really wide set of experimental data. So you use the protein data bank, and then you extract from there pieces of information which allow you to then predict something which hasn't been done yet. So in this case, the structure of a novel protein. And that works as long as you can piece together the parts of that protein or the information you need to predict that from the experimental data which exists. And that may not be the case for all of the proteins that we don't have structures for. And then the other issue is that if you don't validate the result, so if you don't actually end up crystallizing that compound, you won't know for sure. I mean, AlphaFold will tell you how much uncertainty there is with the produced model, but since medicinal chemistry works at a very local scale, so the fold of the protein is nice to know, but the exact shape of the binding pocket is what it's really about for medicinal chemistry, and that's just a small part of the protein. And if there's too much uncertainty in the shape of the protein, the detail of the binding pocket will most likely not be there. So I think there are still probably challenges ahead.
Still some work to do. I'm gonna we're running short of time here, so I'm gonna kind of give Fritiof the last input here. And one thing you mentioned to me when we discussed this in advance, Fritiof, that is kind of topical, is the use of enzymes in synthesis. Do you want to talk about that?
Sure. We all know that chemistry can be quite an environmentally nasty process, and its selectivity is not always at the best. Enzymes can give us, and not can give us, gives us fantastic opportunities to use the kind of reactions that nature does, and frequently with a very good stereoselectivity. And especially immobilized enzymes is a gold mine for synthetic organic chemistry. And I know that at least one of the big players, Merck & Company, has set their vision to skip traditional organic synthesis and use enzymatic synthesis instead. And imagine this cool thing that you can even have more than one enzyme at the time in one reaction. If you can do your chemistry in the same solvent, well, you can do your first, filter it off or spin it off in the most state-of-the-art equipment, and then go on and do the next step without any purification and isolation in between. So that is fantastic opportunities that I see will change the way we make drugs.
Okay, and that will certainly help with things like waste as well. I'm gonna at this point, I'm just gonna thank you both for joining. I hope that this was interesting for those of you in the audience, and I know we didn't get to all your questions, and we will try and answer some of those in the chat afterwards. But yeah, just remains to thank you both very much for taking part, and I hope in the audience you will join us next time for a future edition. So, thank you.
Thank you.
Thank you for this opportunity.